Thank you guys all for coming to the first 4 sessions today. Seeing these talks have been really amazing. We're here for our 5th session, the cancer model session. And after we'll have a short panel discussion for engineering and medicine where we'll talk about future directions in stem cell models and organs on a chip. And so without further ado, I'm going to introduce our moderators, Dr. Jelena Stanislavovich. I definitely butchered that, and Alan Kremiak. Yeah. Go ahead.
Okay, so good afternoon, everyone, and thank you, Naveed, for the introduction. So although cancer tissue engineering is a fairly young, exciting field, it seeks to address one of humanity's oldest diseases. Our first presenter in the cancer model session is Francesca Paradiso. She is a PhD student starting her 3rd year at Houston Methodist Research Institute. Her talk today is entitled 3D patient-derived ovarian tumor model to investigate stroma mechanical and biological contribution in high-grade serous ovarian cancer, HGSOC development. I welcome Francesca to the virtual podium.
Can you hear me? Yes. Okay. Okay, so hello everybody. I'm hoping you have a good time. So heart has entered an era where one species can alter profoundly its environment, like turning desert backwater into one of the most awe-inspiring cities in less than 50 years, like in Dubai. But the environment shapes us unconsciously in more ways than we care to admit. Like the climate influences our mood, the pollution influences our health, and the natural resources might affect human survival. So one of the latest hypotheses on organism evolution is that it has to co-evolve with their environment. And what was really interesting for me is that actually in cancer setting, there was another hypothesis that was really similar to this one that was postulated back in 1889 by Stephen Paget, because he examined something like 700 patient biopsies.
Francesca, we just need you to share your screen.
Sorry, I thought I did it. This one. Okay. So I was just talking by myself. I'm sorry. Okay, I'm sorry for this mistake. Okay, now seems fine. Great, that was a rehearsal. Now we can start. So again, the Earth has entered an era where one species can alter profoundly its environment, like turning a desert backwater in one of the most awe-inspiring cities in less than 50 years, like Dubai. But the environment shapes us unconsciously in so many ways that we care to admit. Like the climate influences our mood, or the pollution influences our health, and the natural resources might affect human survival. So actually one of the latest hypotheses for organism evolution is that they have to co-evolve with their environment. And it was really interesting for me when I started reading about cancer that a similar hypothesis was postulated back in 1889. And actually this Stephen Paget, he was examining something like 700 biopsies post-mortem of breast cancer, and he noticed that the distribution of metastases wasn't random. So he postulated that the metastasis resulted only when the appropriate seed was implanted in its suitable soil. So the seed I'm interested in during my PhD investigation is the high-grade serous carcinoma, which is, let's say, the most common form of epithelial ovarian cancer. And it usually arises from fallopian tube and is treated by a combination of chemotherapy and surgery. The relative 5-year survival rate for stage 1-2 is 92%. So which means that the tumor is still confined into the ovaries and outside the ovaries growing into the pelvis or abdomen. Then 76 for stage 3, so when the cells start growing outside the pelvis into the abdominal cavity, and 30% for stage 4, which means that the cancer cells are already in other organs and metastasize to the liver or the lungs. And the soil which I'm interested in is the cancer stromal environment, which is composed by the scaffold, which is the cancer extracellular matrix, and its inhabitants, which are fibroblasts that get activated, and so they are called cancer-associated fibroblasts. So very understudied in ovarian cancer is the tumorstroma. And as you can appreciate here in following the pathological features of the continuum from normal fallopian tube epithelium to invasive serous carcinoma, the underlying interstitial stroma really changes in its organization. So as you can see here with the P53 signature, the tumor progression, something that it really differs during the two different tumor stages is the collagen type 1, which is one of the main components of the interstitial matrix, the collagen type 1 organization and orientation. So of course, those changes reflected also in the mechanical properties of the tissue, something that is very well studied in breast and pancreatic tumor, which show a higher stiffness properties compared to their normal tissue. And for example, in this immunofluorescence here, you can see some breast cancer cells and how they grow on soft and stiff material. And on the stiff material, you can see that the percentage of invasiveness and the phenotype, so less roundness, which means also resemble a more metastatic phenotype, is characteristic of cells that are grown on a stiff material. And lately, the ECM composition also changes during tumor progression. And there is just one interesting paper on ovarian cancer, and they use omentum. So it means that the tumor already disseminated outside the ovaries in very late stage. And as you can see here, they delineate the matrisome of the omentum. So there is a huge difference in the composition of collagen, glycoproteins, and proteoglycans in high disease score compared to low disease score. So our idea, it was to try to investigate those different characteristics of the extracellular matrix through different models that will investigate the mechanics and the biology mainly. And if we have time, I will also show you some innovative material that we tested before. This is, of course, in combination with the normal fibroblast or cancer-associated fibroblasts with ovarian cancer-derived cells or fallopian tube cells, with the goal, finally, of finding early stromal biomarker for diagnosis and in the end, to develop an anti-stromal therapy. So the first thing we did is going back to the patients and use biopsy tissue to check if actually the mechanic properties are changing in a pathological tissue compared to the normal ovary. And actually using atomic force microscopy, as you can see here, the tissue derived from high-grade serous carcinoma patients of stage 3 is higher in stiffness compared to the normal ovary. So what we did is that we translated those findings into our model system, which is based on collagen type 1, and we use different cross-linking percentage of BDDG cross-linker, like 0.01 and 1%, to reproduce the same difference in stiffness, as you can see here on the side graph. So then we decided to cellularize this scaffold. And we know from the literature that fibroblasts get activated not only during cancer progression, but this kind of phenotype specifically resemble the fibroblasts activated during wound healing. So they need a bioactive molecule, for example, TGF-beta, to start to express some specific marker, which in the wound healing are specifically alpha-SMA, and for example, the secretion of metalloproteases to remodel the matrix. And if they acquire an irreversible phenotype, or in this specific case, in the cancer setting, they start to express, they get activated and start to express the same marker like alpha-SMA, but also fibroblast-associated protein and some metalloproteases. And that's why now it's well known that we call the cancer has the wound that never heals. So we use the same bioactive molecule at first, so the TGF-beta, to activate our fibroblasts. And as you can see here, looking at the expression of ACTA2, which is alpha-SMA, the gene for alpha-SMA. In fact, there is an overexpression of those two markers just if we cultivate the cells on a 3D settings. And of course, they are overexpressed if also the cells are stimulated under TGF-beta, as you can also appreciate from this immunofluorescence here on the side. And this is also reflected in a higher deposition of the matrix, so higher collagen type 1 secretion, and secretion of specific metalloproteases like MMP1 and MMP2. Another biomolecule that like a pool of biomolecule in this case, that we are testing is conditioned media derived from a specific ovarian cancer cell line, which are the SCOF, which is a highly metastatic cell line. And actually looking at the expression of one of the markers that I was mentioning before, FAP, this is the expression of FAP after TGF-beta treatment, which is actually is similar to the same kind of expression level that we obtain after the stimulation of those cells with the conditioned media. The same we found after incubating the cells with conditioned media for the expression of metalloproteases 1 and 2. And so those are the fibroblasts and how they appear in our 3D model. And as you can see, so this is a vital staining, calcine and ethidium bromide is for dead cells. And when those cells are grown in 3D, they actually start to shrink the matrix and to remodel the matrix. And as you can see in the decrease of the diameter, and there are also some dead cells due to the high proliferation. And in this time lapse, you can see that the cells start to remodel this soft material, and there is a shrinking, a very high shrinking of the matrix. Something that we didn't find in the 1% scaffold. So in the stiff substrate, the cells can actually proliferate and they can survive on this scaffold, but they don't remodel the matrix in the same way. So we were interested to see if there was actually a signature or if they recognized this scaffold in a different way. And we noticed a specific signature for metalloproteases 1 that was specific of cells grown on stiff materials. And also an interesting response to the mechanosensor YAP in the cells that were just cultivated on the stiff material. Something that we found also in literature in analyzing one of the patient databases, like MMP1 is one of the markers that really distinguish the metastasis tissue derived from patient from the primary tissue. So what we did so far for this model is that TGF-beta and the conditioned media activate fibroblasts into cuffs, and the fibroblasts can actually remodel the matrix and they respond to a stiffer tissue. So now we are going into the transcriptome and protein array to really detect the phenotype of those fibroblasts. And of course, we will move towards a more humanized 3D model using primary fibroblasts and cancer-associated fibroblasts and co-culture them with the ovarian cancer cell lines and fallopian tube cell lines. So parallel to this, in the second part of my PhD, I'm also trying to reproduce the differences in ECM composition. So like hyaluronic acid in malignant ovarian cancer is very increased, specifically in grade 3 and metastasis. And it's not only its amount, but also its molecular weight that really impacted the tissue. So in high molecular weight is the physiological hyaluronic acid, while the low molecular weight is specific for diseased tissue, and it reduces apoptosis of the stroma, increase the cytokine release, and enhance ACM remodeling, while on the cancer side, the low molecular weight hyaluronic acid enhance the tumor immunity and enhance adhesion and migration. So I'm trying to include now those two different high weight and low weight hyaluronic acid to my scaffold. As you can see from the FTIR, actually there is this peak that is specific for hyaluronic acid, so easily incorporated in my matrix. And interestingly, adding the hyaluronic acid, the low weight, which means the diseased hyaluronic acid, also resembled the mechanical feature that I was explaining before. So a stiffer material related also to a low molecular weight hyaluronic acid. And last, what we also tested before was a novel kind of material, which is derived from Rhizostima pulmo jellyfish. And we created this scaffold material to actually, so the diameter in this case is 98 micrometer. And as you can see from this gel electrophoresis, he resembled the collagen type 1 derived from rat tail and bovine. So the specific signature of the alpha-1 and alpha-2 bands. And it can interact with the 2 different ovarian cell lines, so SCOP3 and OVCAR3. So they interact with the collagen fibers and they proliferate through that. And we already published this paper. So if you are more interested, you can go and check it out. So saying that, I really want to thank my, so I'm in a joint program between Spons University and the Houston Methodist. So I really thank all of the people in the lab that help me constantly, and my 2 PI, so Francesca Taraballi and Dr. Lewis Francis, and all of you for your attention. And to my PhD peers, just in those hard times, or in general, don't forget to stay positive. Thank you.
Thank you, Francesca. It was an interesting talk, and we look forward to maybe seeing more of your work in the near future. So we have a question here from the audience. So Francesca, you see an increase in YAP expression on stiff ECM. Is YAP also elevated in ovarian cancer?
So interestingly, is usually is difficult to assess if the overexpression is functional, because what we are looking now, and is important to see if YAP is translocated into the nuclei, because usually even if it's overexpressed, it can be in the cytoplasm. But so far in ovarian cancer, it wasn't shown that there is an overexpression of YAP. So it's something that we will look forward also in the patient database that we are trying to build at also RNA-seq level.
Okay, and I guess maybe I have a question for you. Since you're kind of moving more towards the beginning of your PhD, what is a most exciting aspect of your work going forward?
So actually, sorry, say it again.
Sorry, that you're most excited about.
And, you know, also moving at half of my PhD from one university to another was interesting, to reset everything and to work with people from a completely different background. So now actually I'm really excited to hopefully get access to patient samples, because my last goal will be to create a 3D model that is more human-based, and it can give, you know, a platform for either drug testing but more a human-related science and yeah, analysis.
Okay, well, thank you so much. I'll let. Oh, actually, we have one last question, maybe quickly. Are you aware of other groups working on a normal human ovarian model for toxicological testing?
So if for normal, it means like the early stages, I guess, if that's the question, I hope so. I know it's very, very difficult, you know, to get non-cancerous, yes. So it's difficult to get specimens, as you can understand, from non-cancerous patients. It can be, you know, other kind of like samples, salpingectomy from patients that have some other kind of issues, but it's different to have like primary samples. Dr. Drapkin is one of the best for, you know.
I'll cut you off for now because we have to move on.
Yeah, okay. I can reply on the question and answer session.
Thank you so much. Thank you. And right now, I'll let Yelena introduce the next speaker.
Okay, so next up we have Julio Alemán, who is from the University of Pittsburgh. And Julio was recognized as MIT Technology Review Innovator Under 35. And he's not only a scientist, but also a TEDx speaker. So I expect a good talk. And he would be talking about how they recapitulate the microenvironment of patient-specific multiple myeloma by 3D printed organoids in order to predict chemotherapy response in patients. And I give the stage to you.
All right, thanks, Yelena. You definitely put a big pressure there for me. Let's get this rolling. So there you go. Do you guys see it? Or you guys see my?
Yeah, we see.
You see the presentation?
You're in presenter mode right now.
Okay. Oh, presenter mode. So let's change What about now?
Perfect.
Okay. So just to break down the name, it is big. We just basically are 3D printing patient samples with a replica of the microenvironment. This work has been done primarily at Wake Forest. Just now that I've transitioned to University of Pittsburgh, we're transitioning this research in finalizing and applying the same technologies for other patient-derived samples here. And so, you know, the microenvironment of the bone marrow is complex. It's still considered a meta niche. That means like there is niches inside the niche, and those are concepts that we have been trying to decipher over the last 4 or 5 years, both in between the microenvironment ECM and also what we're talking about the cellular component and the dynamic of the cellular component. So the idea of where this came down from basically deconstructing first the hematopoietic cell niche, which means how are we're gonna put them all in the same structure, but different cell composition, and see how each one of them behave. So we started first with a bone marrow sample from healthy humans. We got the cells, we got all these cells that come in a bone drive, and then we sorted them. We sorted them in both into sinusoidal, arterial, mesenchymal, and these mesenchymal ones is the osteoblast concentration that we obtained was so low. We use a really fast osteoblast transformation from MSCs. So now that we had all this system, we discovered that you know, under a really basic hyaluronic acid gelatin hydrogel, we can encapsulate these cells, and we encapsulate them into a device. This device, then we put each of these cellular niches into individual compartments, and each individual compartment was randomized as each different experiments were done. And one of the experiments was, okay, let's flow first healthy hematopoietic cells. You see where we can sustain them. Can we sustain these cells for at least a significant period of time? And then let's start doing disease model. So we did, this one was not patient derived, it was cell lines, but we did MOM13, which are a leukemia cell line. And then literally with the lymphoma cell line. And the challenges were major, both about keeping these things alive, keeping them flowing, a universal media aspect. All of these things are already published, so you can address them. But what we learned from this is that as they flow through the device, they have different preferences, especially like, you know, as I mentioned, there's dynamism between the niches, and it's really constantly changing. And so what we discovered is that the leukemia cell, the lymphoma cells are really arterial driven at this stage of it. And as they are arterial driven, we find ourselves with like, okay, now what will happen with our leukemia ones on the cell osteoblastic driven. So a little bit more dormancy when it comes to being remanent in the bone marrow. And then finally, the healthy ones, we really didn't find predominantly localized. There is a lot of theories about the stage of these cells. We discussed a little bit of it, but we learned, okay, we have a platform, we have a hydrogel that can sustain different types of bone marrow derived cells. And so we have a system that can be definitely translational into different cellular components. And those are lessons that we learned from only this. So we said like, okay, what is the next stage? So the next stage is a patient sample. We've been talking about devices all day. I don't know if it has come as a conclusion, but making devices and translational in high throughput devices is still not right there. So we find ourselves in like, okay, which one is the next step? And we're gonna talk about it. Through a collaboration with physicians at Wake, we found ourselves with access to multiple myeloma cells. These multiple myeloma cells, we were actually freshly derived from the patient, like 30 minutes from when they were removed, we were put into the patient, from the patient, we were put into our devices. And we did a different type of research between them. So what was the basic idea? The basic idea is we drive this bone marrow, we isolate all the cellular components, both the myeloma components as the stromal component that we can derive from the marrow sample. This is just a regular marrow, like there is no need for the patients to come back. This is normally part of the regulatory. There's 3 rounds, and we normally get the 3rd one or the 2nd one, depending on the quality. And then we put them in this hydrogel that we have tested. It work on a microfluidic device, right? And so thinking, okay, now we can make a lot of these organoids and then do multiple drug studies. What we have been talking most of the day, drug discovery, personalized medicine, high throughput studies. And so the first thing we find out ourselves is like, okay, we cannot use the same number of cells that we used previously. So we have to optimize the number of cells, as the bone marrow, when it's highly compacted, is highly dense. If you have ever reached into a histology image, it's gonna be highly concentrated. Second, we have on ourselves like, okay, some of these, as they are not, as we're not letting some of these cells mature, or they find themselves derived from a deceased patient, we need to stimulate them. We need to provide them with something that the bone marrow itself, if we don't let them mature, we are not gonna see at all. So we find ourselves with the first candidate of our hydrogel / media component was the use of GCSF to stimulate most of these cell populations. Then we find ourselves like, you know what? Our hydrogel is not enough. We need to find something like, we are not seeing any reproducibility. We were not seeing sustainability throughout time. And so we find ourselves with like, fibronectin is part of the bone marrow component, especially in the diseased patients, in the plasma cells. There's a high stimulation of fibronectin. And so we find ourselves with like, playing with the engineering of the chemistry and realizing that we can actually fix this fibronectin, because when it is just in dilution, it actually goes away in the media, and we don't see a good reproducibility. And so finally, we find ourselves with a hydrogel and organoids components where we have a optimized cell composition, hormonal or growth factor stimulation, plus a microenvironment sufficient factor. But even with all this, we find ourselves with not having a good reproducibility. And all of these experiments are being carried out with patient samples. So we feel ourselves that we are actually not giving back to them, and we're really need to do more work. So we go back to the drawing board, and we just build this massive experiment with some 10 patient samples where we actually try a whole array of growth factors and a whole array of the hydrogel compositions, finding at the end of the day with a composition that was actually highly reproducible and significant when it comes to with the biological aspect of the bone marrow of the multiple myeloma patient, which is a fibronectin impregnated hydrogel, which is fixed, not in solution, plus the application of GM-CSF, so not just GCSF, but GM-CSF, and the stimulation needed from interleukin-6. Thanks to the advances that we have found in chemistry and in our lab, we find ourselves adding the GM-CSF heparin group into the hyaluronic acid that has a characterized heparin group 2. This is commercially available. We find ourselves with a thiolated fibronectin system. The gelatin is always essential. We did some trials with recombinant human collagen. It's just the difficulty with printing, but we found ourselves like gelatin was significant enough, and a PEG as a linker. So we grabbed the multiple myeloma cell samples with the stroma, again, 30 minutes from the patient being removed. We load into a cartilage. We then are able to do these droplet systems. We do the UV light system, and we do putting of media for them to receive a stimulation immediately. And this is important because part of this aspect is that we are being able to do this high throughput with little to non-human interaction besides loading into a syringe. So these are examples in two different types of bioprinters that we have in our lab, where once this is set up, after the 30 minutes of cell processing and loading into the cartridge and hydrogel preparation, we found ourselves printing a 96-well plate with still remnant hydrogel in the cartilage in less than 18 minutes without human interaction. And so as anybody has 3D printed, we'll find yourself that sometimes by the time you're done 3D printing the first well and into the last well, you're gonna find yourself with the first well might be dehydrated or might have some impact. In this case, we're obtaining high cross-linking, maintaining the 3D-ness or the scaffold microenvironment distribution. We find ourselves stimulating immediately with interleukin-6 with the media without having to wash it away or wait to like, oh, let's put this sacrificial media and put it back, and 3D printing the whole platform. And so once we had this is like around 1 year of patient samples, different literature, different work, we find ourselves with finally addressing chemotherapy screens. So we have first these three normal traditional chemotherapy screenings that patients with multiple myeloma go through. You can talk about them. Common denominators are both bortezomib and dexamethasone, which is something that they really want to get rid of. And once we 3D print a whole, these are representative, we can be able to do live death screening, we're able to do metabolic screening, we're able to do histology screening. And then we're starting to see, okay, there's a difference in the impact of the drugs, even though they have really highly common denominators between the three of them. And so we're like, okay, let's do it with a patient, with two patients from the same day, one after another one. So actually, this is a comparison between two patients that came in the same day, 1 hour apart from each other. They were processed almost immediately. And then we find ourselves with a different response and screening to what the drug will impact them, each one of them. And so we find ourselves with a system that physicians can actually find, address personalized therapy and targeted therapy for patients, as the genetic composition of the multiple myeloma is so diverse that it makes almost every patient unique when it comes to this disease. And so the impact of the drug regimen they're being put through is really important to prevent both secondary effects and mortality associated to the drug. And so what do we see in the future of this medicine? So currently we see drug resections being put into tumor biopsies, and then currently these tumors are being missed, these tumors are being fragmented, and then either put it in collagen sponges or xenografts. What we envision with this platform, both because it's modulatory in each of the hydrogel components, but it also being almost medium throughput, is that we can homogeneously distribute all the cell populations, both the stromal and malignant, in a chemically defined hydrogel into uniform organoids. And then in that way, we have a platform that is more reliable and more reproducible when it comes to do drug screening, personalized medicine, and drug discovery. So takeaway home message is we have fabricated a high throughput bioprinted platform, which is capable of using bone marrow derived ECM components for both multiple myeloma and stromal patients. The system provides an in vitro mimicry of an in vivo environment, especially for patient-specific multiple myeloma bone marrow. Then we have an organ construct that are being able to maintain viability for a week. We need to address both the time constraint of this system. Our system is heading into a high throughput platform, both for personalized medicine and drug screening. This is what we have talked, and this is what we are aiming for, both not only in this type of patients, but in all of the other type of personalized medicine approaches we have done. And then the microenvironment network of both the stroma and the disease are important. And these ECM factors play a role that none other platform can reproduce if we ignore them. Finally, this organoid technology can be transferred into many types of malignancies, not only multiple myeloma. So these are the teams. No man is an island, and there's a lot of people and a lot of institutions. So thank you very much.
Thank you, Julio. That was great. I think we have time for a quick question. Pablo wants to know if you are looking to use other more common types of lymphoma, such as non-Hodgkin.
So we believe we might be able to keep this platform for that. So far, now that I'm at the University of Pittsburgh, we are not gonna see marrow-derived or hematological-derived platforms, but the efforts are still remnant at Wake Forest. And so the technology is still being transferred, and we're still in touch with them. So, hey, possibilities are endless, right? The chip proved it as a proof of concept. Now the bioprinted platform has a lot of potential.
Thank you. Another question. Have you considered using gel and May in replacement of the combination of gelatin and PEG-DA?
So a couple of the things about using gelatin and PEG-DA is the dissociation abilities. We have data that I didn't present here with each flow cytometry data. And so we need to release these cells so that when they have been exposed, and so the use of gelatin and PEG-DA allows us to be able to use non-harsh methodologies of cell sorting. But if we manage to find systems that have, click systems to detach or release cells, we might even encourage them.
Thank you. Moving forward to.
Hi. So I'll be presenting the third speaker. I'll be introducing the third speaker. We have Don Trong. He's a second-year postdoc at MD Anderson Cancer Center. And today he's going to be presenting the mesenchymal tissue landscape, a single-cell RNA-seq-based metric of liposarcoma differentiation. I welcome Don to the virtual podium. Don, we can't hear you.
Sorry. All right, there we go.
There we go.
Trying to find the unmute. All right. Thank you for the introduction. So as you said, I'm a postdoctoral fellow at MD Anderson Cancer Center. I work with Dr. Joseph Ludwig. We specialize in understanding differentiation of various soft tissue and bone sarcomas. Today I'll be discussing our progress on liposarcoma. So sarcomas are typically named based on the tissue they histologically resemble. So for instance, liposarcomas arise from white adipose tissue, hence the lipo portion. There are 5 different histological subtypes of interest. In this case, we're looking at a survival plot of the 5. You'll notice I highlighted 2 of them, D-differentiated and well-differentiated. The D-differentiated subtype is a lot more aggressive, as you can see based on the survival plot. The reason why these 2 are highlighted is because they represent a different degrees of differentiation from adipose tissue. So you can see within this histology, the well-differentiated liposarcoma still have some fatty areas in the tissue, whereas D-differentiated liposarcoma, which we believe D-differentiates towards more of a cancer-like, or cancerous phenotype, has less fatty tissue, but more nuclear characterization. And we know that most or many soft tissue sarcomas have high expression of YAP-TAZ, as you can see within this slide for well-diff and D-diff. There's a high nuclear staining of YAP. Again, we see high nuclear staining of TAZ for both well-diff and D-diff. And we know that YAP-TAZ pathway regulates cell fate and differentiation. And in fact, YAP-TAZ is considered as the molecular rheostat of a cell. So whenever cells sense changes in the stiffness of the environment, YAP-TAZ will respond. So within this normal context, as we can see here, mesenchymal stem cells will respond on a stiff environment by localizing YAP-TAZ into the nucleus and priming the cells for osteogenesis. On the other hand, when we take a look at cells on a soft matrix, YAP-TAZ will stay outside of the nucleus, but priming the cells for an adipogenic phenotype. Now, when we take a look at a cancer, in the context of cancer, YAP-TAZ is often localized only in the nucleus, as we saw in the slides before. And we have 3, downstream effects: proliferation, cancer stem cells, and survival of the cancer, which are all hallmarks of cancer. And one thing of note is taking a look at the cancer stem cells, knowing that YAP-TAZ can cause the cancer stem cells to arise, we can think about how D-differentiated liposarcoma may have arisen from well-diff liposarcoma due to YAP-TAZ expression. So that brings our hypothesis where we believe that deregulation of YAP-TAZ prevents liposarcomas from maturing to a normal adipocyte phenotype. And this is why we're observing that there's high expression of YAP-TAZ in the D-diff liposarcoma. So we have our sort of model that we imagine mesenchymal stem cells differentiated towards either adipocytes or osteoblasts. Bear in mind, these are just 2 of the lineages for the scheme. There are multiple lineages, but we'll be looking at just adipocytes and osteoblasts. And based on the hypothesis, well-differentiated liposarcoma, which are more fatty in nature, would show up near the tail end of the trajectory. Whereas D-differentiated liposarcoma, less fatty, more stem-like, more proliferative, will show up higher up on the trajectory, closer to a mesenchymal stem cell phenotype. So this brings up some big questions that we want to answer using our hypothesis. So can we generate an in vitro model to understand YAP-TAZ regulation in context of liposarcomas? And will diff and d-diff map onto the landscape as we hypothesize? And what are some deregulated gene networks that we can learn that prevents liposarcomas from maturing or sustaining the differentiation blockade, such as YAP-TAZ pathways? So here's a workflow of our experiment. We take mesenchymal stem cells and we grow them on either soft stiff hydrogels or high stiff hydrogels for approximately 5 days, followed by an induction medium towards either osteoblasts and adipocytes. And we'll be utilizing single cell RNA sequencing to frequently sample the cells as they undergo the differentiation process, so we can learn about how the transcriptome changes, what are different gene regulatory networks that are occurring over time. Now, the first thing is we're going to take a look at just the first portion, where in this case, cells, specifically mesenchymal stem cells, are only undergoing changes based on a stiff environment, either low, high stiff, or tissue culture polystyrene. And can we use this model to understand normal mechanosensing and learn about dysregulated pathways in liposarcomas? So first, we tested for the YAP response in mesenchymal stem cells. And as you can see in the figure, YAP has higher nuclear localization as stiffness increases, so which suggests that these cells can sense the stiff environment. And further downstream, what we want to know is what are the changes in the transcriptome. So as I mentioned, we use single cell RNA sequencing to profile these cells. Now here we have our what we call the single cell atlas of mesenchymal stem cells on substrates of varying stiffness. We have a UMAP, which basically each dot is a single cell representing their transcriptome, and their placement on this map denotes their similarity between cells near them. So for instance, most of the cells that were cultured on tissue culture polystyrene ended up on the left side of the map, whereas cells cultured on high stiff gels are mostly in the center, and cells on the low stiff gels are on the right side. So as you can see, the cells are placing themselves on this map based on stiffness. Now we can take a look at some of the gene expression such as YAP1 and TAZ, and we can see that for the most part on the left side of the map where cells are on the higher stiff substrates, we see upregulation of YAP. Not as much expression change in TAZ, which we also observed in the immunofluorescence. We didn't see a lot of change. TAZ may not be as robust within mesenchymal stem cells. On the other hand, we do observe ACTA2, which is alpha smooth muscle actin, which is well known to be highly upregulated for mesenchymal stem cells grown on very stiff substrates. Now, taking a look at some of the genes that show up for softer substrates, we see MKI67 proliferation, as well as CD73 and CD90, which are the two mesenchymal stem cell markers appearing slightly higher on the soft substrates. So now these are just single genes, but we want to understand some of the larger gene expression programs that are downstream of YAP-TAZ. What are the regulatory networks that YAP-TAZ are controlling? So we can utilize a algorithm called latent Dirichlet allocation, which essentially creates topics for us based on the expression of genes. So inputting all of our cells and if all their gene expression, each topics can be defined by how correlated different genes are. So for here, we can observe in our heat map here. So you can see we defined approximately 12 topics. And then we have our 3 conditions, low, high, or tissue culture polystyrene. And you can see that these topics, some of these topics are regulated by changes in stiffness. And we can annotate some of these topics using annotation tools such as BioPlanet. And we observed that topic 3 is highly enriched for YAP-TAZ stimulated gene expression programs, as well as TGF-beta regulation of ECM. On the other hand, topic 5, we observed that it's enriched for cell cycle, DNA replication, and M-phase pathway. So the question is now that we understand how some of these topics or these gene expression programs are being regulated with a normal model using MSCs, how would it look in liposarcomas, which are cancers? So we have some RNA sequencing data. This is bulk RNA sequencing data of well-diff and D-diff liposarcoma. remember, dedifferentiated liposarcoma is the more aggressive variant. We projected the topics onto the single cell data. So here are our 12 topics again. And first thing you'll notice is topic 3, which is the YAP-TAZ expression programs are highly elevated in D-diff liposarcoma. On the other hand, we also observed topic 5, again, the cell cycle related programs are highly elevated in D-diff liposarcoma. And when we look back at the normal response that we had on MSCs, what we should have seen is that with increasing stiffness, MSCs actually downregulate cell cycle responses, whereas they upregulated YAP-TAZ response with increased stiffness. But when we take a look at the context of cancer, these two topics are more or less entwined.